Okay, we're starting to see plasmolysis. This is actually the same leaf that we were looking at before. I removed the cover slip, removed as much water as I could with a tissue, kind of sucking things up from the side. And then I added the salt water and then re-established the thing. And one of the things you see is you're definitely seeing a lot of active cytoplasmic streaming here. The reason why that's happening is because you're seeing more, this has been exposed to more intense sunlight, in the sense of the light from the microscope. So these cells are getting more and more active. However, one of the things I'm hoping you can see here is that the cells are, the chloroplasts are now much more likely to be in the center of the cell. So if you see a lot of these cells in the middle of your field of view, you don't see the chloroplasts out on the outer edge. You see them all kind of bunched up in the center of the cell or on one side of the cell. And the reason for that is that as the salt water is encroaching into that, through that cell membrane, the fresh water is trying to pump out. The cell can't control that. And as the water gets lost, the cell membrane shrinks and shrinks and shrinks. A really good example is kind of right in the center of the field of view, sort of about 1/3 of the way up, you see a cell where all of the chloroplasts are sort of clumped in the very center. That's a really good example of a cell where you can almost see the cell wall that's retreated. Sometimes if I change the light level, I don't know if it'll make much of a difference here, but I'll try to change the light level. We can get a little more of a depth of resolution. Now remember, we're looking through layers of cells here, okay? So I'm trying to kind of work through the focus. Yeah, the focus isn't working really good at this level. The camera's really struggling to make sense out of what it's seeing. So I'm gonna go back and increase the, chuckle butts, that's not what I wanted to do. I'm gonna increase the light level. I don't know if I can find this now without switching it back down. So I'll have to move this back. Boy, I really lost that. Okay, so here's my leaf, okay? Again, I'm going to focus this up. And you can see the little edge there, those little toothy leaves, which it looks like it's completely smooth, but then when you see it like this, you're like, oh, there's a lot of stuff going on there. So I'm gonna pop this puppy onto high power. And now I'm gonna focus it up. And again, I think you can see how these chloroplasts are now clumping in the center of, there's some around the edges for sure, but they're also kind of clumping in the center of these cells. You can kind of see them retreating right there. There's some good views. Now, obviously, there's still some cells that are still doing pretty good because you're seeing a lot of cytoplasmic streaming doing on some of these upper ones. You see that one up on top. A couple of them on top are really doing well. So if I try to pull up on that focus, you can really see cytoplasmic streaming really taking place, going great guns. Those cells are either inside the leaf entirely, which is possible, or they will be, so they're less exposed to the salt water, or they're up next to the cover slip and aren't getting as much water. Meanwhile, the cells that are down deeper in this slide, which I'm trying to focus on for you, but I'm struggling a little bit, you can see they're much more clumpy. And the chloroplasts are much more in little bundles inside with the cell membrane kind of pulled back from the edges. Notice again you have your cell wall. The cell wall is those very distinct hard lines. They're sort of clear or with a contrast of gray that you're seeing on here. You see if you stand back, the big overview, you see a kind of a dark gray grid, almost looks like a fence, like you're looking through a chain link fence. That is the cell walls on the top layer of cells. And then I'm now focusing on to a lower layer of cells where you can see, again, the cell walls that are more clearly highlighted. They're going slightly to the upper left, to the bottom, the bottom right. They're just ever so slightly angled that way. So if I don't want to do that. If I pull the field of view back a little bit, it's not really helping you to see it very well, but you can see kind of how I can focus on these different layers of cell walls and chloroplasts. But again, the cytoplasmic streaming, which is pretty cool.